I'm so pleased to have Dr. John Catanzaro, CEO of Neo7 Bioscience, with me today. Thank you so much.
Thanks, Dr. Michael. I appreciate you being here. Thanks for inviting me. I'm looking forward to our talk together here.
So, what you have put together is really fascinating. And in regards to cancer, people fighting cancer out there, we are learning more and more the importance of personalized care. And obviously, that not every cancer is the same. You can have 10 individuals, all triple negative breast cancer, and still each one of those cancers are unique. How did you go about that? The technology you guys are bringing is phenomenal in regards to that.
First of all, a technology background on the Air Force, and I also in the aviation industry for about almost 12 years before I went into medicine. And then I got accepted into medical school. I graduated, went into clinical practice, and started a pretty good sized cancer center outside of Seattle. We had 8 practicing physicians plus pretty large staff, and we did 25 years worth of work, clinical work, and research in the area of understanding molecular signals, protein signaling, and how those protein signals behave in different type of disease types. And cancer was the primary disease focus, right? So we actually started all of our work on cancer. And we learned a lot from that. Multi-disease within a disease is what I usually say. You wind up getting so many variant signals. And like you said, it's not the same in every person. So it's usually what is variance between person to person. And one person can have the same exact diagnosis, but their design criteria is totally different than the other person. So we usually call it's an N of 1 design with personalizations. And that's the reason why we have so much success at Neo7 Bioscience in these designs, is because we are looking at the individual molecular expression. Everybody it expresses their processes of function differently. There might be some things that are similar. Not everything is similar.
And so here you have an individual that has cancer, and now you're wanting to create something that is unique to that individual, like a therapy that can then specifically address that cancer in that individual. How can you do that? What technology are you guys using to do that?
Technology was developed over a significant amount of data inputs during those 25 years of clinical practice. And when we formed the company in Neo7 Bioscience in 2019, we were a lot further ahead. Our platform was developed with contracted labs that we worked with at Dana-Farber Cancer and things like that. So we had some pretty smart people that I corralled as a team, and we wind up identifying what we were finding in patient samples that we were isolating. Like, in other words, we saw certain type of patterns in patient samples that seemed to point in the direction that if we gave them specific type of peptides that were derived from those proteins, that those patients got better. But we weren't able to figure out exactly what those targets were until we got together at Dana-Farber and figured it out. So then when we started putting names to the faces, so to speak, then we were able to really craft the engineering appropriately. But it took us in 2019 about 18 months, along with my co-founder, who's a molecular biophysicist. He's a Johns Hopkins alumnus and very smart person. He and I got together. And it's very interesting because we met on LinkedIn, and he said to me, I saw your work. I'm interested in your work. I have something for you. Let's get together and talk about this. So we got together, and that started a whole new paradigm movement into development of a complete molecular profiling using patients sampling that would get them to a whole different paradigm in designing personalized treatment. So what we do is we take the samples of the patient, which is blood, urine, and tumor tissue, and that blood, tumor, and urine give us a caricature of data, high definition data, that allows us to profile some very sophisticated signaling in disease evolution. So we know the hallmarks of these diseases. We know the hallmarks of cancer expression, and we're learning all the time, right? Currently, there's about 15 different cancer hallmarks. And we have been able to figure out where the ranking is in certain protein-to-protein interactions that affect the cancer and cause the resistance in the cancer. So like a lot of patients that we've designed for are stage 4 resistant cancer types, and they wind up beating their disease because of the fact that we're hitting those targets directly with their own personalized peptide engineering. And so we're really excited about it because we've moved about 3 generations from the time that I first started.
And yeah, so what kind of results have you seen? You mentioned some stage 4 that you've seen some incredible results. Can you give some samples of what you've seen? People that did not fare so well with traditional oncology, and they were in a dire spot, and they implemented this therapy, and where are they now, so to say?
Yeah, so there's many cases. One that comes to mind right off the bat is a nice lady who, stage 4, she had prolific disease everywhere. She had it in her lungs, she had it in her bone, she had it in her liver, and it wasn't looking pretty. The PET scans looked terrible, and the physician that we were working with saying, hey, I think this is your best shot for you to beat this diagnosis here. And they were doing everything they possibly can integratively, right? They were doing a lot of intravenous supportive treatments. They were doing things like EBU. They were doing intravenous anti-cancer therapies. They were supporting the liver. They were doing a lot of different things, wonderful things, right? But it wasn't getting any better. It was still progressing. So we got a design made for her. And within 3 months' time, this woman, her disease began to regress significantly. And by 65% within the first 3 months. And then within, I think it was something like 9 months, she was totally clear. Her PET scan came back totally clear in every area. And we have another patient with triple negative breast cancer. She's alive now, going on 12 years. She had a very large size. I was probably about a 19 centimeter small melon type tumor on a mastectomy site. And she was bleeding from this tumor, and she had to be transfused every 3 days to stay alive. And the conventional docs gave her zero hope, saying that you got to get your affairs in order. You only have like probably 6 months to live at best. And if you continue to bleed like this, probably a lot less than that. So she wasn't taking that. She came over, she was referred to by another physician to us to get a design made, and we got the design made. And I'm telling you that within a period of 6 weeks, that tumor started breaking down in large pieces. It started disintegrating like amazingly so. She went back to her oncologist and said, hey, I look at the size of this tumor. It's like nothing like it was 6 weeks ago. And she stopped bleeding. She didn't need to be transfused anymore. This woman is alive today, going on 12 years. And it's just amazing. And we've had other cases other than breast cancer like this. We've had colorectal cancer cases where I have a wonderful young lady. She has 5 children. At the time, she had disease from head to toe. She was receiving every type of conventional treatment you can imagine. Toxic chemo regimens. I wasn't sure if we were going to be able to do anything with her, but we worked with her for about 18 months and got her out of that tailspin. She's alive today and celebrating her family. She's doing really well. The cases like that, we've had cases like resistant brain tumors this way. We've had stomach cancer cases, bladder cancer cases. And now we're also moving into active disease of chronic illness, where these mystery illnesses that are not having easy things like Lyme disease, neurodegenerative disease. So we're moving into that realm now because we're seeing such success in cancer that we know we can have success in targeting other disease types. And so we're moving into personalized treatments in those realms too.
That's really exciting. That's incredible. And obviously this is non-toxic. This is supportive. This is nothing that is destroying anything in your body when you're doing this type of therapy, right?
That's absolutely correct. And it's very supportive. It's interesting when you're using personalized approaches like this and personalized engineering, what you wind up getting is not just the actual attack on that disease type, but you get the regenerative new cycle cells that develop with better healthy signaling platforms to help regenerate better healthy function as the person starts recovering from this. So you get rid of that, you eradicate the disease, and you move into a resilience movement. And peptides and personalized peptides that are targeted are just amazing because they actually facilitate that type of change.
Okay, so here you brought in a word, personalized peptides. What does that mean in this process?
Yeah, personalized peptides simply mean is that when we do your molecular analysis from your blood, urine, and tumor tissue in cancer, we isolate those top targets that are causing the problem and the resistance and the disease, right? So we want to help to refocus the molecular signaling paths and get the immune system to toggle into the problem, target it, get rid of it, and help to regulate better, right? So these personalized peptides are constructed specifically to address those targets that are causing resistance. And how they do that by signaling ability. So they help to re-signal the protein. So proteins that are called unstable proteins, they're called aberrant proteins. That means that they are very unstable and they cause an erratic type of a signaling communication from protein to protein. So if one protein is not signaling correctly, its downstream effects to other proteins that are supposed to keep signals will also be affected by that. So when we talk about personalized peptides, we're talking about those specific peptides to those specific peptide protein targets that are going to help facilitate better signaling exchanges with the next succeeding cell lines. So that actually means that you get rid of the old signals and you're allowing for the rebirth of new signals and healthy cell generations.
And how does that help somebody with cancer? So here you get better signaling going on, and how can that resolve cancer?
It resolves it by, first of all, correcting the DNA patterns, number one, helping with the transcription. Like, in other words, every protein that's made has a DNA order, and it also has an RNA typing ability, transcription. In other words, the RNA types out the message. So the DNA dictates what message is going to be typed. The RNA types out the message. And if the message is healthy, it's going to create better function or balanced function, right? In cancer, if that message that's being typed, like in the case of a mutation, it's going to create havoc. It's going to create a crisis type of event. That means that the directions of the cells go awry. That means the cells don't know what to do. So they wind up proliferating very fast, and they wind up creating adhesive signals to each other, which creates tumor formation, right? So not only do you have cells circulating, and this, I'm trying to simplify it, but not only do you have cells circulating in the system, or cluster cells or cluster tumor cells circulating in the system, but you also have a tumor environment that starts to form in different places, right? So the immune system becomes tolerant of this, and then it allows for things to happen that shouldn't be happening in the body. So what we're doing with the personalized peptides is we're reprogramming things. We are saying you're not supposed to be circulating. You're not supposed to be getting stuck in that environment over there. We're saying that you need to be out of this environment because you are not supposed to be in the system, not tolerating self, right? So we use the ability of the body to have a better interrogation in the immune system to target that offending agent, which it now recognizes as being the cancer cell or the cell with a bad signal, and then to go after it with the various defense mechanisms like T cells, natural killer cells, CD4 helper regulator cells, CD303 dendritic cells, APC machinery type structures, which means antigen presenting capabilities to get rid of that offending agent, so to speak. And those personalized peptides are what create that whole milieu of activity to allow for that transition to occur. So if you don't have something that's going to hit, now see, chemotherapy, what it does is chemo is going to have a direct killing action, but a lot of the times what it does is it creates more resistance, because then cells that actually generate after are then resistant to the agent that you originally gave. When you're talking about personalized engineering, it doesn't work that way, because those signals propagate over time. So that means that the new cells that develop will actually have a healthier regulatory mechanism, whereas the chemotherapy doesn't do anything like that. It's a direct killing agent with no reprogramming ability at all. And the same thing for radiotherapy. Radiotherapy actually is targeted radiotherapy can be very advantageous in dropping down or debulking tumors or creating an environment that's a lot more stable so that you can hit it better, but it's not going to change the scenario. It's not going to change the bad signaling platform. So if you don't have a signal change, you might have reduction, but over time you might find that the resistance even becomes greater because of the fact that the signals haven't been corrected.
So it sounds to me then, so these signals, they allow the immune system to more effectively kind of see where the weaknesses are within this tumor, and then also to these kind of microclusters or the microenvironments, the immune system is then able to see which signals that are healthy, that are not healthy, and then correct the unhealthy ones.
That's right. You hit it right there. And the other thing is that if you can stabilize the transcription and the protein signaling, you're going to be doing a lot in the system. Any kind of stability that you can lend on DNA to RNA transcription to proteome and protein stability, that means that protein interface is not going to be all shifty where it's creating this chaotic oscillation that doesn't really serve well for homeostasis, because what it does is it creates a dynamic shift in favor of disease rather than in favor of function. So a healthy function, right? There's function, like in cancer, there's function on the disease side of encouraging unhealthy function, which we don't want. So we want to stop that signaling progression. We want to facilitate stabilization on good functional proteins, and we want to get rid of mutation activity, and any kind of aberrant proteins that are going to produce that result. And we have the ability at Neo7 Bioscience to detect those proteins that are causing those specific issues. And our system is able to categorize the top targets that are the highest in the evolution of that process, so that they are the focal points, because you can't hit every target. But if you actually hit the toggle targets, the ones that are actually the ones that can switch and matriculate down to string relationships or sister proteins, those are the ones that would matriculate a better action on all protein stability, because there are main heavy hitters that create better protein stability on other proteins, right? And in the cancer evolution, you get to see, and for instance, everybody knows about BRCA1 and BRCA2 with breast cancer, but what they don't know is they don't know that there's a BRCA complex, which actually is called the BRCA MRN. It's a BRCA MRN complex that's responsible for regulating cellular proliferation and DNA repair. And if you have other signaling genes in that platform that's created that machinery, right? Everybody's looking to BRCA1 and BRCA2 as the troublemakers. They're not always the troublemakers. The ones that are troublemakers are the ones that are in that machinery that create the havoc in signaling to create more DNA disrepair and a lot more cell proliferation signaling disturbances. These are some of the things that a lot of people don't realize, but at Neo7 Bioscience, we know it and we see it in the data.
And obviously, if you're then clearing out the unhealthy DNA that creates these kind of aberrant signals, and then correct the signal mechanism so the DNA is healthy, strong, creating, and the RNA, is healthy and strong. Now we're creating then healthy tissue, versus then cancer tissue.
That's right. Absolutely. That's right. That's what we're facilitating. That's where we got this lead movement into, because we understand that dynamic. And believe me, with the new information that's coming in all the time, one of the benefits that we have is we have collaborations with Elsevier and companies like Sericel, Elsevier. So we understand knowledge graph integrations, we understand neural networks, we understand how data placements are supposed to be, and how to get the data to actually come up with an end product that's going to solve problems. So we're not just talking about creating a data lake where you're just putting all this data in and you're trying to just become a storehouse for data and have data as an asset. Data is not an asset if it doesn't have an output engineering that solves a problem. Neo7 Bioscience has taken that data, real patient data, fleshed it out to create an end output engineering that solves the problem.
And one of the terms is that this is like a personalized vaccine against the kind of the antigens that are presented, or the components from the cancer that the immune system can check out and then create an appropriate immune system response.
Mm-hmm. Yes. And so from that standpoint, just to tell you about peptides, and I've been working since 1998 in this peptide, especially in the cancer space. And I've got to see some of the advances in some of the different research, like, for instance, Catherine Wu and the work that she's done over there on neoantigens. And of course, the term neoantigens is a fashionable term to say, oh, because they're new, they're antigens that are seen on different cancer expressions and things like that. But you know what? That does an injustice to personalized peptides in a way, because personalized peptides are not just about antigenic responses. Personalized peptides about creating edits and signal responses. So it's not just about eliciting T cells or NK cells. It's about correcting, like you said, aberrant signals or aberrant signals that are really the core and the crux of creating what we call proliferative disease. And so we say that, yes, you'll have a neoantigenic effect to calibrate the immune system, but you're also going to have a protein stability effect in creating better signal interfaces. And that's what we're really interested in, is the signal interfaces more than we are in just recruiting immune system. Because you can recruit natural killer cells all day long, but they may not be directed against the tumor. They could be directed against other tissues and not be specific enough, right? So unless you actually have a target to beat and you have a direct signal to have as your cargo, then you're not going to have the desired results that you want in these type of cancers, especially when we talk about N of 1, means single individuals that fight their cancer in a different manner. Not everybody fights the cancer in similar manners. They might have similar mechanisms, but they may not have the complete same manner in which they fight the cancer.
And this way, because we are getting the data from the individual directly, we are then able to obviously present an end product, like you're saying, that is then unique and specific for that individual in a manner that individual would fight that specific cancer that they're battling.
Yes, that's correct.
Because we have this new field of immunotherapy, CAR T cell, and so and you just made the point clear that we're not just looking at the immune effect, the heightened immune system response. We are also looking at the intelligence, the communication, the signaling, so that the immune cells know exactly where to go, what to hit, where the weakness is, and then do target response on that, rather than just do a blanket increase the immune response.
Yes, you see the difference with CAR T cells, and I've been involved in looking into different areas of powering up CAR T in a better way to make it less toxic and more effective. CAR T, when you have a random antigen that you're focusing, like for instance, they were using CAR T in some prostate cancers to try to direct certain type of traffic by taking a specific synthetic cargo to a location by using CAR T. Now, the reason CAR T has some negative collateral effects is because it's not selective enough, it's not sparing enough, and it creates an off-target reactive action. And the reason for that is that it's not truly personalized to the person's own molecular signature. It has components of foreign nature. You can't mix the foreign in if you have to have a more direct related pattern. So if you're just going to use a synthetic portion that you can call maybe pseudo personalized, it's not really truly personalized. It's still a foreign entity. It's still something that elicits the possibility of an off-target toxicity associated with. So you wind up seeing a lot of CAR T having more off-target reactions because it's largely donor, it's largely antigenic, and it's largely non-specific to the person. So in that particular case, then you're opening it up you may have some favorable effects, but your unfavorable effects far outweigh your favorable ones. And the research actually shows that across the board in many CAR T studies. Now, how do you make CAR T better? You make CAR T better by having your carrier cells or your shuttle director cells with a better intelligent cargo system. So if you have a better cargo that you're carrying and it's more related to the molecular expression of the patient and specific to the disease evolution, then you're going to be able to direct that traffic a lot more effectively. So if you put your hands up to stay stopped, you're not going to have stragglers trying to run the yellow light, right? So
What I'm hearing is that essentially, what Neo7 Bioscience are doing, these peptides, these personalized peptides, it becomes without the collateral damage because it becomes specific. And then in addition to, over and above that benefit of the immunotherapy, in addition to the very specific, you are then adding a component of better regulation. So instead of then producing disease tissue, you are then producing healthy tissue. And then also in addition to the communication that's going to increase within the body.
that's very true. And of course, we get to do it better and better as we learn more and more about how, what the variances are, the expressions are. So we learn from case to case about how to make the adjustments more from a specific precision standpoint. So from every case, we've learned that there's similarities and significant differences, but at the same time, our bottom line is protein to protein interactive abilities. So when we want to be able to work, because you, as with your experience, the dynamic actions of proteins that are keeping functional gain of function gain, I don't like to use that gain of function, but functional integrity is essential in keeping the body intact and warding off disease. If you have good, strong functional integrity and you maintain that strong core, then you're going to have less aberrant signaling coming in those proteins. And the older we get, as we age, the more risk we have in aberrant protein signaling happening. Because the thing is that's a natural progression. But here's the thing, is that once you actually graft in this new type of engineering that's specific to the patient, has the ability to perk up memory systems that may have been lagging down because of various type of aging cycles. And that's why we're also working in resilience too. So we're covering resilience because we understand the disease hallmark of cancer, but we also see the correlative relationship between the cancer and the aging expression. And you can see there's quite a bit of overlap there that the research bears. And we are picking up on okay, so we see that if we address these signal changes and we help to what we call help with better interrogation, better surveillance, better defense, better regulation, and better adaptation. That means we're covering the sector of being able to move into a better mimicry, if you will, a natural mimicry of the body, something that might have been lost, right? That we're trying to really harness to say, remember, you're supposed to do it this way. And if you never did it this way, this is the way you should do it, by allowing that engineering to take hold there, right? And we do that by either mitigating, regulating, potentiating, and influencing the protein path. And every cell has, you have to know the individual expression that's going on with the person. That's so crucial. And you could use different things like augmented peptides to help facilitate better activity and better performance and better responses to reducing age-like actions in the body. But really, the best way to do it is to find the hub strategy first and then build your other strategies around the hub strategy. That's what we usually say at Neo7. You have the hub, and then you can build an intelligent system to support your hub. And we find that's where the greatest success winds up happening, is the docs that we work with are just doing it that way, and they see amazing results because of that.
Yeah, and at the end of the day, cancer has a hard time to live in a well-functioning, communicating, strong structure. So by supporting healthy tissue function, then we're going to win the battle. And it's fascinating when I do these kind of dry blood samples from individuals, and you can then see the tissue integrity. So you can see how much fight that's in the tissue in regards to cancer. And then if there's not a lot of fight, that then cancer has an easy time to move forward. There's not going to be a lot of resilience. And by obviously creating that resilience, you're able to restore that fight.
Absolutely. Yeah, I've heard it said, I don't know who said it, but it's an algorithmic relationship to where cancer has to overcome algorithmic odds to be able to sustain in the environment. Like the body is constantly adapting and shifting against that activity. It's always in favor of a resilience movement rather than a succumbing to disease. But there is a point of a crossover point where, like you were just saying, that it winds up becoming a higher load more in favor of what the cancer progression is, rather than on the regenerative side of resilience. And I think that the thing is that when we understand that the body is it's a miraculous organism. It's able to do things that we can never imagine. No, it's really interesting. I don't know if you've seen on LinkedIn, but they had this LinkedIn thing that was taking people's heads and trying to put them on healthy bodies. It was called head transplants. And I'm just thinking, I'm sorry, guys. I don't know. I don't know where you're going with that, but this is not what Neo7 Bioscience does. Neo7 Bioscience is really honing in on what makes sense in the engineering process to help facilitate wellness. That's what we do. And when we're talking about trying to get your body to overcome the odds and move on the algorithmic side of beating that disease, that's what we're about. We want to get you to that algorithmic 10x and greater so that you can get to that space of having healthy resilience. And a lot of the times you have to address the disease first that you're working with. Sometimes people want to put longevity on top of a moldy wall. It doesn't make any sense to put resilience on top of a moldy wall. You're going to have to get rid of that mold and mildew and get rid of all that before you can. Yeah, in other words, they want to do performance peptides on top of thinking that they're going to re-energize their body when they've got a root cause that they've really got to address. So we're about understanding what the root cause is, getting a person through that process with personalized design to a better platform of beating that disease, and then moving into a resilience engineering that can actually sustain them for longevity and a healthy longevity.
And we talk about performance type of peptides like CGC, epimorelin, MOTS, those type of things. And so, by not having the core signaling component corrected, then adding that on top to aberrant signals doesn't make any sense.
No, it doesn't. And you really got to address that core. It's getting to that root. It's the old paradigm was healing comes from the inside out, from the top down, and in the opposite order as the symptoms appear. Like, in other words, a lot of the times these illnesses are driven so deep into the energy of the person, you don't realize how deep they are. It's not the end of the world, people. It's not. It's actually a good thing that we understand personalization and precision, because we can get down to the core, get your constitutional energy up to where you are in a more balanced state, but you're doing it intelligently. Like you were saying, Dr. Michael, it's about being intelligent about how you do this, because you can't layer a high performance picture on a engine that is blowing a lot of oil.
Yeah, no, that I can't agree with you more. So a couple of things that are out there that are similar in my mind, but I wanted you to tell me how they differ. You have something that's out there, it's called like SOT, then you have dendritic vaccines. How is this different, than what's out there?
Yeah, now SOTs are the supportive oligonucleotides that are RNA components, or they could even be DNA components. So you can have your oligonucleotides, which are your backbones of your nuclear material. And there are certain type of sequences that can be very constructive in being able to facilitate a better, what we call transcription rate. And a lot of the times you can achieve that on the short term, but you can't do it in the long term. A lot of these SOT therapies are known to have short-term signaling and a higher decay rate, which means that they wind up dropping off too soon. And then you can't. And the other thing is, as we saw with mRNA vaccines in COVID, we've actually seen rogue effects that occur. Like, in other words, unpredictable protein transcription effects that were undesirable, which we didn't want to see. It's translation that's gone awry, basically. It's translation that's uncontrollable and it's gone chaotic simply because of the signaling base is unpredictable. So you not only do you have a rapid decay, but you have an unpredictable transcription that can occur by using certain things like SOTs without better direction. Now, if you used SOTs in conjunction with personalized peptide directives, you're going to have a lot more stabilization on your SOTs because your SOTs are going to be able to direct traffic a lot better. They're going to be able to have a better messaging intact system with probably more predictability and less rogue reactions, right? This is what we find. Now, so what they've been doing now in the research environment is they're trying to do what they call peptide oligoconjugates, which means they combine peptides with oligonucleotides. Instead of calling them an SOT, they call it a POC. So a POC is something that can help to direct, it's like having a DNA barcode onto your peptide, basically. And you're directing the traffic that way, right? So not a bad concept. It's just that it's in its very early development. And we're learning more and more about it too, because we want to be able to see if we can capture the best of that technology as well and turn it into what we call microprotein assembly, because you can direct your microproteins that stabilize your main proteins. So we're figuring that part out too. But the thing is that how it differs is that it's like taking the nuclear material, putting that in first before you actually take the assembly material, which is your peptides or basic building blocks. Your peptides are known as your basic building blocks. It's so they actually help to work with a better natural mimicry rather than what we call a supercharging entity that can create undesirable effects that you weren't planning on. So that's the difference there. Dendritic cell therapy is great too. They have their places. Dendritic cells have to be directed. If they're not directed appropriately, then they're just dendritic cells. They're dendritic cells that help with what they call antigen presenting capabilities, what we call APC machinery direction. And if they have a good intelligent cargo to rely on, then they're more effective. But if they have a really lousy one, they don't do very much. They really don't. It's like putting a dendritic cell vaccine without an intelligent cargo system. It doesn't work very well. Same thing with mesenchymal exosomal cells. You can use them if you have good intelligent cargo in cancer, like these peptides, because they can be a very effective carrier, similar to CAR T, but even more acceptable than CAR T is. And they'll be more effective if they have intelligent cargo programming on them. If they have a good cargo load, so your shuttle trucks that are going to take it to the main areas that need to be delivered to with the right effects. Whereas dendritic cells, like I said, if they don't have the right programming done, then they become very ineffective. And we used to work with CD303s and 304s when we were back outside of Seattle. We actually cultivated them from the patient's own. We magnetically separated them, we expanded them, and we cargo loaded them with the peptides and gave them back to the patient. And they worked very well. They worked excellently. But if you gave dendritic cells alone without good cargo, not as effective. Not as effective. Same thing with MSC exosomes. Given without intelligent cargo, they're not going to be as effective in cancer or any other type of targeted therapy. They would be effective if you're just going to give them generally for regenerative longevity, because they're going to create that regenerative effect, but they're not going to be specific in these other disease types. You know what I mean?
So when you talk about cargo, you have them and dendritic cells, not right cargo, exosomes, not right cargo. And then you mentioned the peptides, as the cargo. So I take it that it's these very specific peptides that we identify through the process over at Neo7 Bioscience.
Yes, that's correct. So if you identify the right cargo, then you're going to be able to charge up all of your carrying systems. So your MSC exosome systems, your dendritic systems, your natural killer cell systems, your CD8, CD4 interactive systems, your CD25 regulation systems. So like, in other words, we're talking about there's so many different signaling platforms in the cytokine arena and the cell signaling arena for defense. And if you have the right cargo system along with it, you're going to encourage the right defense posture, the right regulation posture, and the right adaptation posture. So if you have something that's too heavy on defense, then you, like in cancer cases, when you have an aggressive tumor lysis type of activity where tumors break down very fast, you have to have a component in your cargo that's going to be able to address a regulatory posture, so that you can create a mimicry signaling that's going to give the body the ease so it doesn't create that shock effect like you see in tumor lysis, right? So in order for you to do that, you got to understand the posture of T4, CD25, and interleukin 10, and some of these other cytokines that are responsible for creating that regulatory posture. So you have to have defense posture, you have to have regulatory posture for a better adaptive movement forward. And you're doing that so you're helping the body with its ability to have its own constitutional energetic mimicry to be able to move in that direction. So that mimicry, by what those peptides do, facilitate a better propagating signal forward to allow for a better balanced movement to beat the disease. Just get that heavy defense without no regulation. And how we learned that was we had many cases in Seattle that we treated that had very heavy tumor burdens. And when we gave very heavy defense-related peptides with no regulatory peptides, we had a very high tumor lysis burden. And we found that these patients they would go into a shock. So we had to really address that complication. And we did. But as we learned, we said, okay, how do we create a better dynamic effect in the body to have more of a natural progressive regulation so that you don't have such an intense shock effect to the body, right? And these peptides, when they're correctly designed, do that. And so we find that the peptides that are being generated for these patients based on their molecular expression have the ability to correct, like I said, those string relationship signals of the other proteins that are creating these different signals. And a lot of the times we'll also see that, okay, if this patient has a very heavy tumor burden, then we need to have more regulatory movement in to create, number one, a better regulation in support of the defense. And two, in the tumor microenvironment, sometimes you get resistance because of a heavy macrophage imbalance, right? So you wind up getting a macrophage slimy layer on the tumor, and you can't break through that barrier and penetrate it. So you actually have to then have a balance between the right regulatory posture and the right defense posture to get into that tumor. So our designs have what we call an RGD penetration capability, which means that it penetrates the tumor. And it also has what we call a TAT signaling, which means it's a trans-activating factor signaling that allows for sensitivity of the cell signaling to alert the immune system that it needs to go into that space. So that's powered up on the peptide. So it means the peptides are not just naked structures coming in. They're structures that are programmed with TAT and RGD capabilities to move into that space, right?
That's incredible. That's incredible. So what does it look like practically for a patient, to go through this process?
It's not really that difficult. It's just a matter of getting a blood sample, a urine sample, which is a random urine, and those are sent to our labs, and getting the tumor paraffin block, or what they call FFPE tissue. We can also isolate it from fresh tissue too, but most of the times fresh tissue is not available because they use it to put it into the paraffin so they can probe it further later. So it's blood, urine, and paraffin tumor tissue or fresh tissue if you have it. It could be also any type of fluid that comes from ascites or fluid accumulations in the lung or whatever in the peritoneum. We can sample those and do sequencing from that too. But most of the time it's blood, urine, and tumor, right? So it takes about 6 weeks to multiplex data from those samples. So you just send those samples into the lab. It takes 6 weeks to get the multiplex data back. In other words, the data build. And then it takes a week for the data build to go through the Neo7 platform. And then the interpretive design with the report comes out within that week. So it's about a 7-week process there and about a 3-week process to manufacture the peptides.
Okay, so we're looking at about kind of a 10-week process before we're able to introduce that into the individual.
Yeah. And sometimes we can expedite if the lab is on point and they're really moving along well. We might be able to get that down to 8 or 9 weeks.
Okay, okay. And so now the patient has then the product. What do they do with that? What does the administration of it look like?
Yeah, very simple. We work with the doctor. So Dr. Michael will be working with you. We don't market to individual patients. This is all directives from the doctors that are treating. And the therapy can look like 3 times a week intravenous support with IM injections, along with any other integrative therapeutics that are supportive with that. And it's a process by which, if the cancer is pretty aggressive, we're going to be really treating it on the front side pretty aggressive for the first 6 months with no breaks, because we want to pull a person out of that tailspin. So you're going to be receiving weekly injections all the way through combination IV-IM. You can have some other supportive peptides that are anti-cancer peptides and regulation peptides, like PNC 2728 combo that we have at Neo7 Bioscience. We also have a thymulin-nutlin peptide adjuvant that stimulates the immune system. So we have that too. A lot of docs are using that in conjunction with personalized peptides to facilitate that robust defensive posture. And then within 6 months, we should see a much different picture. And if we do, we wind up saying, okay, you might be able to put a 3-week break in between. So the first 6 months, if it's pretty intense, is going to be more piggyback all the way through, so that we can get the desired molecular signal exchange that we want. Now, patients that don't have as nearly as, I would say, aggressive type picture, then you can be a little bit lighter to where you can do more of 2 to 3 injections a week with your other supportive measures, and you can be a little bit lighter. You can have a break in between. The other thing is that the personalized peptide therapeutics can work very well with targeted radiotherapy, conventional immunotherapy, and also with therapeutics. And as a matter of fact, there's a lot of literature that supports that the personalized therapeutics using peptides can ameliorate negative effects of these therapies as well and create better functional salvage from that, right? So in other words, you want to be able to maintain as much functional integrity, keep your body strong through this. A lot of times people are beaten down pretty well with chemo, radiation, and other therapeutics, and even with conventional immunotherapy, because they just blast you away with that. And here's the thing, is that we have found that we've actually had cases where, like in targeted radiotherapy, the peptide sensitized the tissue, where the targeted radiotherapy actually got every piece of the area in question, all margins and everything, to where that tissue responded so favorably because of the fact that those cells were sensitized in that area. We have been finding those reports that doctors have been giving us as well. Not only that, but I'll just share this. We've had patients that have come to us that were originally a single cancer diagnosis, and then all of a sudden they were laden with another cancer diagnosis. So we wind up designing and redesigning to address 2 different cancers in the same person. And we've actually done that successfully with several cases.
That's
I think,
exciting.
yeah, yep.
And are there certain type of cancers this works better on than others? It seems to me that it should work equally well, because all you're doing is that you're working on it from a functional standpoint rather than the specific tumor type.
That's correct. The point of that is that when you're doing this N of 1, which is personalized, you're getting down to the very core, I would say, disturbances that are creating that disease process. So they work with the most resistant cancer types, and it doesn't matter what type of cancer it is. We've had brain tumors, we've had pontine-resistant tumors, glioblastomas that are coming, and we see these designs that are coming out that are pinpointing the exact movement. Here's the other thing, is that we've worked with a major research institute, and they were doing a design on a pontine brain tumor, and they were just isolating out one target. And they said, can you guys run this data through on your platform and see what you get? We came up with 12 different targets. And they said, whoa, okay. We were only planning on hitting one target. I'm sorry, but it doesn't work that way. I think you're barking up the wrong tree if you're just thinking that you're going to hit it on one target. I'll give you an example. There was a research study that was done a few years back on 26, I think, patients that had glioblastoma, GBM, very resistant. And all of them were in an advanced stage. So what they did is what they call a neo or neo pseudo personalization. So what they took the common expressions from all the patients and they tried to say, okay, this is unique and common to each one of these patients. So we'll isolate these out because they're common, right? And they only looked at mutations. They only looked at somatic mutations. They didn't do anything else. See, at Neo7 Bioscience, we do mark these gene protein markers in excretion from the urine. So you can have an absent mutation in a gene. As a matter of fact, we just had a case that I just reviewed today, a BRCA2 in a germline. The mother died from this cancer, breast cancer, BRCA2, and her daughter expressed no BRCA2 in the somatic mutations. She expressed the BRCA2 in the germline mutation, and she expressed a hot BRCA2 with abundance in the urine. So that means that she's excreting it in the absence of a somatic mutation. So in that study that I'm telling you about, the 26 patients with the brain tumor, and I'm trying to correlate this so you can see the relationship here, is they only focused on somatic mutations. They didn't focus in on any excretion protein related to the actual disease process. And they admitted that in the design, because all those patients, those 26 patients, they all expired. All of them did. And they concluded that we shouldn't have been just looking at somatic mutations. We should have also been looking at protein targets that were disease related in the absence of somatic mutation. And we actually saw that plainly in this breast cancer case where somatic mutations negative, hot excretion of BRCA2 in the urine, and a positive germline. And very rarely will they go to a germline to determine an actual target, because it's a lot of the times they'll say, well, it's unreliable because it's an inheritance pattern. Again, especially when the mom died from it and the sister got the same diagnosis and now the other sister has it.
That's fascinating. Because it's exactly like that. What happens, the oncologist, they order them to see where the genetic mutations are, and
right.
It's done then on the tumor tissue. But here you're saying that, yeah, it may not express in the tumor tissue, and you will then find it in the urine or the blood. And by then correlating all three, you're then getting a much clearer picture. And in addition to that, then you're looking for signaling defects as well over and above these genetic mutations.
Yes, and how it correlates is important because you can see the values of the one doesn't really correlate with the other, but you can see the persistence in the prediction system once it runs all the way through the pipeline. And you're saying, whoa, I would have never, you would look at it and you would say, okay, everybody's thinking along the lines of how conventional medicine trains you. PD1, CTLA4, CD27, LAG3, they go all to these checkpoint targets. And then they'll say BRCA, and then they'll say JAK, and then they'll say other type of genes, KRAS, which is another one, P53, which is in everything. So you wind up looking at all these different protein gene fashionable names, but really they're like just a little tiny bit of the puzzle because of the fact that they are like the ones that everybody's been focusing in on conventional therapy in research to be targets. And what they do is they wear out their welcome because the cancer knows its workaround ability. It knows how to get around. The DNA switches are going to happen somewhere else. So you better pick up on the signals that are hitting those downstream triggers, because that's where your battle is right there. And when you're correlating the data in its completeness, like you said, Dr. Michael, you're looking at it from the standpoint of saying, I need to understand this in a greater measure, because you'll miss something if it's being excreted in the urine in abundance. It's a hot biomarker. And if you miss it, you miss a vital part of the programming that can change the picture.
And that's the thing, like you're talking about cancer, they have all these different escape mechanisms. And so you have all these doors that are open, and where it can then go to continue its own proliferation and also the metastases to move into other areas. And if you
Yes.
just shut two of those doors, it still has maybe 10 other doors to go through. So that's why
Hmm.
it's so important to be able to look at more of these signaling aberrations and mutations to be able to close all those doors to make it really hard for the cancer to exist within the tissue.
Yes, and then more of success in being able to produce a propagating signal effect in favor of long-term healthy living and disease-free. Because once you train those cells, and those cells, that DNA is intact, the RNA transcription is intact, the protein signaling is functional and strong, and you're getting new cell lines that are producing that over and over, and you're facilitating that activity, then you're going to keep an intact organism much healthier for a longer term. And it's really crucial to understand this. And look, we're learning all the time. As technology improves with our ability to understand more real-time development, and this is not some futuristic thing. This is available right now. When I was in clinical practice, I've treated thousands of patients in clinical practice, and 8 practitioners, they saw many of their patients too. We collectively corresponded and communicated on all these patients. But we took 500 of these cases to show that we could make a difference with no hope scenarios. And since we've been in business in Neo7 Bioscience, we've actually created over 200 designs with resistant cancer and other active diseases. So we're gaining a lot of data that is going to be useful to help people overcome the most hideous things, including these post changes that you see with this COVID vaccine. We've actually had a number of cases already that have shown some pretty significant severe aberrant signaling caused by the vaccine related injury. And the process is simple. You submit your sample, it goes to the lab, it gets done within that 6 to 7 week time period. You get your peptides made, 9 weeks, about 9 weeks on average, right? And you wind up starting your therapy, and you're on your road to a better regenerative type of health, a better stabilized health, much better quality of life. And that's what we've been seeing across the board with so many of our docs that we work with.
It's really wonderful work you're doing, Dr. John. I so appreciate you taking the time to explain the beauty and the benefit of this new technology. I wouldn't say new because you've been doing it for a long time, but it is new to, I would say, to a lot of people out there. And yeah, it's exciting. For me, it's exciting to be able to offer this kind of therapy at my center. And I know it's phenomenal type of therapy, and to really give hope to a lot of people that do not have hope. So thank you so much, Dr. John.
Oh, thank you so much, Dr. Michael. It's wonderful being here. And look, anytime you want to have me on again, I'm happy to answer any questions or whatever you think. I'm available. So we look forward to working with your patient base, and we're appreciative so much of having you on board with us, because I firmly believe that the paradigm of health is changing in favor of these personalized treatments, and you're going to see people having some amazing transformations in their life.